Hello and welcome to this podcast on cell signaling part 2. In part 1, if you remember, we talked about the 5 major steps of cell signaling. We said the first one is the production and release of a chemical signal, which upon its release is going to bind to a receptor protein. That's the reception part of cell signaling. That receptor protein, we said, could be on the cell surface, as a membrane receptor, or it could be inside the cell as a cytoplasmic receptor. Binding of the signal to the receptor would trigger changes inside the cell, and in the case of membrane receptors, would result in a signal transduction pathway that would result in the formation of several relay molecules, leading to a cellular response. The activation of this signal cannot go on forever. It has to be deactivated. And the deactivation process is as important as the activation process itself. And the deactivation is a way to regulate the chemical signaling and as a way to deactivate it and terminate it. So we'll start with the definition of signal transduction. And as I mentioned again in the first part, signal transduction involves a process by which an external signal is converted to a different type of signal inside the cell that is amplified thousands and thousands of times. So if we take the example here of epinephrine as the signal, which acts on an adrenergic receptor or epinephrine receptor on the membrane, which is a G protein coupled receptor, resulting in the formation of this second messenger here, cyclic AMP. That cyclic AMP, when it's produced, it activates protein kinase A, which in turn is going to activate a phosphorylase kinase enzyme, which in turn will activate a glycogen phosphorylase enzyme. That enzyme would act on glycogen, which is the storage form of glucose, and hydrolyze it into glucose 1-phosphate, which is then dephosphorylated to produce glucose, which will diffuse out of the cell and flood the blood with glucose. This is what we see here happening from the production of cyclic AMP all the way to the activation of the enzyme. The last enzyme here is the signal transduction pathway. And we can see here the actual amplification. From one molecule of epinephrine, we produce 20 molecules of cyclic AMP, leading to the production of 10,000 molecules of glucose. That's the amplification process. So the signal transduction, it happens inside the cell, and it involves membrane receptors. The two types of membrane receptors that are involved or that work through signal transduction pathways are G protein coupled receptors, like in that case here, epinephrine, as well as protein kinase receptors that act through a cascade of phosphorylation. So in that protein kinase receptor, the signal transduction pathway involves the autophosphorylation of the receptor itself, which leads to the phosphorylation of different kinds of protein. In that case here, the first one is RAS. The activation of RAS would lead to the activation of another enzyme, a RAF, and then RAF will activate MEK, and then MEK will activate MAPK, and then MAPK can go into the nucleus and activate transcription. So this is another example of signal transduction pathway, and in that case here is that of a growth factor that would result in the formation of certain proteins through activation of transcription.